Thank you for the introduction. Greetings, everyone. Today, it's my pleasure to share some thoughts on the role of GDUFA research in promoting complex generics. First, I would like to briefly discuss why, how, and what do we do with GDUFA research program. What is the purpose of GDUFA research program? The overall purpose is to promote generic development and facilitate the assessment of generic drugs. How do we conduct research? We have been conducting targeted research projects through internal and external collaborations. Once a project is done, what do we do with research outcomes? We use the knowledge to facilitate development and/or revision of product-specific guidances. We use the knowledge to help address technical questions received in regulatory inquiries to facilitate generic development programs, and when ANDAs are submitted, we use the knowledge to support ANDA assessment and final approval. In today's talk, I will primarily focus on what do we do with the research outcomes by elaborating on the details through a few examples. In the next two slides, I will briefly discuss how we have been using GDUFA research for supporting product-specific guidances. In general, research has been helpful throughout the life cycle of a PSG. Before and during development of new product-specific guidances, an in-depth analysis of potential research gaps can be helpful to refine the types of studies we may recommend to support a BE approach or the kind of research we have to do prior to being able to develop a product-specific guidance. And once a PSG is posted, we can still identify research gaps to help support potential revisions to types of studies or methods that may be used for a particular bioequivalence approach to further improve the BE recommendation. In addition, research can also be helpful to facilitate the assessment on any particular tools or methods that may be used to conduct BE studies. One good example to show how research can be helpful throughout different life stages of a PSG is the one on levonorgestrel intrauterine system. In this PSG, we recommend an in vitro ex vivo combination approach, which was not recommended before. This approach was developed mostly based on the understanding of formulation design as well as potential in vitro in vivo drug release mechanisms. During the PSG development, we identified the need for developing a new statistical acceptance criteria for the recommended in vivo ex vivo study. So we internally collaborated with the modeling division as well as biostats to address this scientific gap through a modeling approach. Once the PSG was posted, we realized there are still remaining scientific gaps, such as assessment on qualitative Q1 sameness of silicon elastomers. Accordingly, we have been doing research to better understand the impact of silicon elastomer characteristics on product performance to improve our current understanding, so we'll be able to develop scientifically sound approach to help assess the Q1 sameness of a silicon elastomer in a proposed generic product. In addition, we also recognize the real-time in vitro drug release testing for this product is challenging. So we have been doing research to explore the possibility of using an accelerated in vitro drug release testing to replace the need for multi-year real-time data. Next, I will switch gear to discuss how does research support generic drug development. We publish our research findings in peer-reviewed publications and hope they can serve as a valuable resource that generic company can use to support their generic drug development program. In addition, the improved understanding of complex products enables us to provide more effective feedback to generic applicants while regulatory inquiries, such as controlled correspondences and pre-ANDA meeting requests. So in the next 2 slides, I'm going to give you a few examples to further elaborate on these points. In this slide, I'm going to give 3 examples of publications containing readily adaptable methods. The first paper is about reverse engineering the 1-month Lupron depot, published by Dr. Steve Schrinderman's group at the University of Michigan. In this paper, the group developed analytical methods for facilitating reverse engineering of Lupron depot as well as other similar PLJ-based products. Through the study, we obtained better understanding on qualitative sameness of complex inactive ingredients such as PLJ polymers as well as gelatin. The second example is about analyzing ophthalmic suspension particle size distribution. This was a new method developed by the FDA lab at the Office of Testing and Research. This method was able to accurately measure API particle size without interference of inactive ingredient. One thing we recognized about characterizing complex products is that the properties of complex products are often interrelated and not straightforward to measure or compare. So this is the kind of scientific gaps we're trying to identify either during product-specific guidance development or through the information we received in regulatory inquiries, and we then try to develop targeted research to help resolve some of these technical difficulties. And the third example is about separation of PLG polymers when used as a mixture in one drug product based on differences in lactide glycolide ratio. This work was done by Akina Inc. So throughout the 3 examples I like to highlight, we are trying to proactively identify complex products as well as scientific gaps associated with those, and then develop targeted research to come up with new methods that may be helpful. But one thing I like to point out is by no means we are requiring generic companies to follow the exact methods. So how we view those methods is that they can serve as a potential starting point, and companies can adapt the methods as appropriate based on their program. Before moving on to discuss how research may help ANDA assessment and approval, I'd like to give you a couple of examples to show how we have been using research to enable efficient communications while regulatory inquiries. The example products I'm going to focus on are injectable suspensions with micronized particle for short-term use. So a couple of years ago, we revised the PSG for catalog 1040 triamcinolone acetate injectable suspension. That revision was done based on our understanding of the critical formulation characteristics and their impacts on product performance and drug release. So ever since then, we have been continuously working in the area of injectable suspensions to identify products or categorize products under different considerations, and trying to see what are other products that may be able to also have an in vitro only approach to support bioequivalence. So methylprednisolone acetate was one of the products we identified, and in the meanwhile, we also received proposals from several different generic applicants to develop alternative in vitro only approach for establishing BE of their generic products. So based on the improved understanding obtained through our internal and external research projects on injectable suspensions with particle size in micro range, we were able to agree with a proposed alternative in vitro BE approach last year and updated the PSG to ensure timely communications with all generic applicants. Lastly, I'm going to share some insight on how does research support ANDA filing, assessment, and approval. As discussed before, one area we have been heavily investing is the characterization of complex polymeric excipients. So the knowledge we obtained through various research projects enabled us to develop appropriate acceptance criteria for determining qualitative Q1 sameness of complex polymeric excipients to support ANDA filing. Research has also been utilized to provide technical support during ANDA assessment to obtain approval. I'm going to give you one example in the next slide. The example I have here is cyclosporine ophthalmic emulsion. This is a complex ophthalmic product, and this PSG was the first one in which an in vitro only approach was recommended for supporting bioequivalence of a complex ophthalmic product. Ever since the development of this PSG, FDA has been conducting lots of research to investigate various aspects, including the impact of process parameters on critical quality attributes, how to properly characterize global size distribution, rheology, drug distribution of the product, as well as how to determine in vitro drug release from the emulsion, given there is no compendial method available. This long list of publications really highlight the research efforts FDA has invested in cyclosporine ophthalmic emulsion to be able to have the first generic approval earlier this year. To summarize my talk, first, I hope through the examples I have shared today, you gain some insight on how FDA has been utilizing GDUFA research program to support projects that are designed to improve the understanding of complex products issues, which can promote generic drug development and approval. Secondly, new methods developed through GDUFA research can serve as a starting point to facilitate generic development. Third, we recognize the early identification of specific scientific and regulatory knowledge gaps is critical for generating targeted research outcomes. We have been trying to proactively understand the reference product to determine any under-investigated areas that could be critical for generic development, so we can develop targeted research accordingly. We have also been accumulating feedback on practical challenges faced by generic applicants during development while regulatory inquiries. Therefore, it's really important for generic applicants to communicate with us about your technical difficulties in a timely manner. With that, I would like to thank for your attention, and I'll be happy to address any questions, comments you may have during the panel discussion. Thank you.